All right, it's my pleasure to introduce our third speaker today. Abdulmalik is a PhD student and a graduate research assistant at Auburn University under Dr. Tim Bruce and Anita Kelly. Currently, he is investigating the effects of humic substances and similar feed additives as growth promoters and immunostimulating agents against polymicrobial infections in channel catfish and tilapia aquaculture. His research activities have been on dietary stimulations and optimization for overall fish health management using natural and cost-effective additives. So thank you very much, Abdulmalik. You can take it away.
Yeah, thank you, Dr. Phelps. Just like Dr. Phelps said, my name is Abdulmalik, and I'm a graduate student here at Auburn University under Dr. Timothy Bruce and Dr. Anita Kelly. And today I'll be presenting to you the project that we did last year at West Alabama, Greensboro. So we basically are looking at the effect of humic substances and protease complex on the growth microbiome of channel catfish in an outdoor system. So to start with, channel catfish, as most people already know, is actually the largest aquaculture species in the United States, with about $437 million in sales in 2023. And even though at that, one of the major big issues in the industry has always been bacterial infection, which has actually amounted to a very significant loss in the catfish industry generally. And those are basically virulent Aeromonas hydrophila causing multi-Aeromonas septicemia, Flavobacterium coriae causing columnaris disease, and also Hydrazella ictaluri causing enteritis septicemia, or basically hole in the head disease. Looking at this, sometimes last year, but it's actually a 7 years wide study conducted by Abdulrahman et al. In West Alabama, where they're actually looking at causes of fish loss. And what they come up with was bacterial infection amounts to about ×3 of non-bacterial causes, fish losses causes in West Alabama. And as seen from their results, also we have VAH, columnaris, and also ESC being the 3 most significant bacterial causing disease in channel catfish in West Alabama. And they further concluded that about $30 million has been, an estimated loss in West Alabama, and $550 to $600 per ton is spent on medicated feed, which is very significant for farmers. So similar study was actually done far back in 2019 by Peterman and Posadas, where they also came to a conclusion that about $6.9 million is being lost to bacterial disease, basically in Mississippi. So looking at this, our lab was trying to look for alternative treatments, because some of the treatments we have basically now majorly is antibiotics, and that has been a major issue because of antibiotics resistance bacteria that have been developing, and also because of the limitation with antibiotics. We have very limited of them in the United States. So we try to look at dietary options, and what these basically do is it improves the immune system of fish and helps them to be able to fight these bacteria better and improve their survival significantly. So we started looking at humic substances and protease complex. Humic substance is actually an extract majorly from water, from soil, from most organic sources generally, and protease complex has been as a result of bacterial fermentation. So we try to incorporate these two into channel catfish diet, and we fed it to them sometimes 2 years ago in an indoor system, and we found significant protection and also significant increase in immune system, and also potential for growth in a low protein channel catfish diet. So basically, the protease complex was 175 grams per ton, and also the humic substance was 0.5 pounds per ton. And this significantly improved resistance against columnaris disease, which I'll be talking about next. So in our results, which we have already reported in a paper published last year, when we challenged the fish that were fed immunostimulated diets against F-Covey ALG530 in an indoor challenge trial, we found out that these 2 immunostimulated diets actually reduced mortality significantly compared to an high protein diet or even a low protein variant of the same source. And as you can see, we have the blue and the green line representing the HS and the PC. I'll be referring to it as HS, humic substance, PC, and protease complex for the rest of this presentation. And I have the green and the blue line, so please take notes as I keep presenting. So that we decided to escalate this trial and try it in an outdoor system, and also look at this influence on gut microbiome generally. So talking about gut microbiome of fish generally, we can see some of the factors that influence it, which can be temperature, which can be culture system, maybe indoor or outdoor system, which can be the ontogeny, the lifestyle of the fish at any particular stage can determine the kind of bacterial community that will be present in the microbiome. And also salinity can also drive that, as well as dietary type that is given to the fish can also determine the microbiome source. So for this presentation, I'll be focusing majorly on the dietary parts and what we were able to supplement to the diet and how we got the results that we had. So yeah, prebiotics, and dietary supplements generally, when given to fish, they modulate the microbiome community of fish and they significantly improve growth. And also a lot of reports have also saying that they improve the immunity and also survival of channel catfish when fed probiotics and dietary supplements. So that led us to our objective and experimental design, which was done last year in West Alabama, which was to examine the gut microbiome of channel catfish. They were fed humic substance or protease complex in an outdoor system. And that was the commercial farm where the trial was carried out up on a pond levee in 12 tanks. And we have the inlet and the outlets there just to let you see how we ran the flow through, and we try to minimize our culture space. So channel catfish were fed 3 diets, basically the humic substance diet, the protease, and also a control diet, which was not supplemented. We did sampling at 30 days, 60 days. We basically collected fecal for microbiome, blood for serum activity, lysosome activity, and also kidney and spleen for gene expression. So we did a 2-way and over to compare the differences across the globe. So this was where we carried out, and this were our tank, and we fed twice daily. We had 3 treatments, 4 replicate and 25 channel catfish juvenile per tank. And some of our routine activity was to pick up mortality and to necropsy those for potential disease that interacted during the culture period. So looking at our sample processing, because I'll be focusing much more on microbiome parts, we collected fecal sample at 30 days and 60 days. And we collected it in RNA liter, and we actually preserved that in -20. We extracted genomic DNA extraction using the PowerSoil Kaijin kits. We sent those out for sequencing to Zymo Biomics majorly, where they prepared the library and also the multiplexing, and they did that on Illumina NextSeq. When the sequencing were done, they did the multiplexing and separated the samples, and they sent that back to us, and that's where I began my read processing. So for the processing, I did that in R using the Data2 pipeline. And the first step was to do the replication and identify unique sequences. And I performed a chimeral check, filtered the reads, removed the primers, and also constructed an ESV table using the merged reads. So the merged reads, the merged and trimmed reads were also actually used to construct a phylogenetic tree, and also taxonomy assignments using the Silver RDP database. And all of those were actually combined and loaded into Phyloseq in R. So my taxa, my ASV table, my sequences, tree file, and also the metadata. So in Phyloseq, basically what I did first was to do quality control by removing chloroplasts, reads belonging to chloroplast and mitochondria, and also trimming any reads that is less than 1,000, which basically I found 3 of those, which belong to my 3 treatments, so which makes my normalization very good. So for normalization, I did rarefaction and also cumulative subscaling for downstream analysis. And for within sample comparison, which is alpha diversity, I did community richness, evenness, and channel diversity. And for beta diversity, which is across treatments comparison, I did Bricotes and unweighted unifrac distance metrics. And I also did a relative abundance plot to look at the composition of bacteria out there. So to show you what the growth looks like, after 60 days, we found significant growth in protease complex and humic substance fed channel catfish. They had better conversion ratio and also tank biomass gain. No difference was seen in relative weight gain, but the K factor, which is the condition factor, also we saw that the humic substance did way more better, and they were more robust compared to the control or the protease fed diet. This was also very consistent with our former indoor trial. And the big thing was the survival, where we found out that the channel catfish, they were fed protease complex and humic substance in this environment, the outdoor environment, significantly improved their survival compared to the control, which is kind of interesting and also gives the direction for our next study. So looking at the microbiome comparison at 30 days and 60 days, you can see that At 60 days, it is kind of very similar, but at 30 days, we see a little bit and a slight variation under the phyla classification. I also did the general classification just to spread things out a little bit. So going forward into alpha diversity, at 30 days and 60 days, we did a two-way repeated measures ANOVA. We found treatment differences, no time differences, and no interaction. At 30 days, no differences, but at 60 days, we saw that richness reduction in HS-fed dietary group. And looking at evenness on the other side, we saw the significant increase in HS-fed dietary group at 60 days, but at 30 days, it was similar. So what this alpha diversity comparison can tell us was that even though these two immunostimulated diets improve growth, but at 30 days, immune substance seems to have a very great effect at 30 days, but when fed beyond 30 days, it might actually reduce alpha diversity, which might be translated to some kind of stress that we haven't examined yet. So looking at beta diversity, which is the across sample group, the first thing I did was we got us the similarity index and using the multivariate differences in centroid to compare treatments. So we found treatment differences, no time difference, and no interaction between time and treatment. And at 30 days, we saw that the protease complex basically have a distinct effect on species composition in channel catfish after 30 days. Whereas at 60 days, they all look similar. And also, following up, we did the unweighted unifrac, which basically compares the bacterial presence and absence among group. And we can see that immune substance seems to influence the structure of microbial community in terms of evolutionary relationship, because unweighted unifrac considers the phylogenetic relationship in its comparison. So once again, at 30 days, immune substance seems to be differently associated compared to the control or the protease complex diet. So we collected blood also at 30 days and 60 days for serum lysozyme activity. And after doing a two-way ANOVA, we found significant interaction, which made us spread things out by analyzing at two different time points. And at 30 days, we saw that both protease complex diet and immune substance diet significantly improved serum lysozyme activity than the control. Whereas at 60 days, it was similar, even though it maintained a very high lysozyme activity. So this also still agrees with our microbiome results that at 30 days, this two immunostimulated diet may have a better influence over a very short period of time than when continuously fed. So we also took kidney and spleen for immune gene expression. So firstly, I'll be talking about the head kidney. We analyzed 3 pro-inflammatory gene and 1 anti-inflammatory gene. The 3 pro-inflammatory gene we analyzed were IL-1 beta, IL-8, and TNF alpha, and the anti-inflammatory gene was the TGF beta. So we actually found no treatment effects, no time effects, but we found significant interaction. And after analyzing at different time points, we also found no treatment effect. But one thing that was very obvious here was that protease complex from 30 days to 60 days seems upregulated at 60 days. So which tells us that protease complex might have some potential at the head kidney and also might have responded to some kind of inflammation happening in the cultural environment. So when we analyzed the spleen at 30 days or 60 days, we found no difference in time, interaction, or treatment. I would like to talk about the immune substance, as we found nothing in the spleen and kidney. This was also very consistent with our last study. And one of the things we've noticed is, even though it's not significantly expressing some of these immune genes, it still proved to be very, very effective in microbiome composition and also in the survival of channel catfish. So this might probably seem that immune substance has some other effects in some other part of the fish, and also might some studies have actually proven that it has some antibacterial effect. So if it actually has an antibacterial effect, it might not necessarily need to actually present itself as a inflammatory driving factor. So in conclusion, we've seen that protease complex and immune substance influence the gut microbiome and improve channel catfish growth and overall health in the production system, not only the indoor trial this time around. And immune substance also seems to improve alpha diversity after a short time application, but when applied beyond that, it's reduced alpha diversity. And these two immunostimulants also has the potential for enriching bacterial channel catfish bacteria in the gut. And future direction we have right now planning is to see how continuous supplementation of these immunostimulants, both protease and humic, can affect channel catfish over a long period of time, and also to play around with the dose response of humic substance over time so that we can optimize it for growth and for health. And my activity in the next maybe couple of years would be to start looking at how this humic substance and protease complex might drive polymicrobial infection in channel catfish, after we've seen that they've proven to be effective in an environment faced with multiple pathogens. So I would like to acknowledge my advisors, Dr. Tim Bruce, Dr. Anita Kelly, and Dr. Alan Davis, Alabama Fish Farming Center crew at Dr. Kelly's lab, my fish lab members here at Bruce Lab at Auburn University, and funding from USD NIFA and AES AgriSeed. And lastly, I would like to show you my reference that I've cited throughout this presentation. And thank you all for listening. And I would like to take your questions now.
Great presentation, Abdulmalik. Thank you very much. Interesting project.
Thank you.
Folks, we have a minute or two for questions. If you have them, feel free to unmute yourself or drop them into the chat.
Hi. Oh, I saw the text question. How
Go
should
for
we
it,
do that?
Benjamin.
So, hi, Abdulmalik. I actually am doing a humic project up here in Canada with some salmonid species, and I had two short questions. I was really curious why you chose to compare the humics to the protease complexes, because that kind of seems like two different approaches to immunostimulation. And then my second question was, one of my biggest kind of bones to pick with the humics is that you have this guy over here making a product and this guy over here making a product. How do we all know these are comparable, and why did you choose your humic substance product for your test?
Okay, so yeah, thank you so much for that question. So I'll start with your last question. So for the humic part, we're actually looking at cost-effective additives, actually, and that was actually what led us to the products we took, not specifically for any type of chemical composition, because we've not done that yet. And I'm also aware that we have multiple humic substance products out there, and that is actually what is driving my next project, to start looking at some of the composition of this humic substance to see how the composition actually drives some of the effects that we've seen. And that would make us to decide what products as what composition and what would perfectly treat for growth and immunity that we're looking at. And for the second question, comparing humic substance and protease complex, in this particular trial, we did not do it for any reason, because we know that those two were produced under different conditions and they were meant to do something different. So, but we just decided to look at how these two immunostimulants might drive channel catfish, and comparing them together was not because we thought they were the same. No, they were actually very different, and yeah, that actually didn't drive that. So it was just comparison just made to just see which did better than which.
Thank you so much. We should get in touch after, because we could probably use our substances and compare their structures. I have a few ideas on how to do that, but
yes,
yeah,
thank
you so much.
Yeah, thanks. Yeah, thanks.
And we have one last quick question here in the chat for you. Did you notice any change in the fecal characteristics influenced by the diet?
Fecal characteristics about the diet?
Yep.
No, actually, not for now. But my next project is also actually looking at the microbiome of this fecal at different time points, and also considering the microbiome of the diet itself. So maybe that would give me more information about the diet itself in relation to the fecal composition.
Sounds good. Always more work to do. Thank you very much for your presentation.
Yeah, thank you.